Students, this is our sixth lecture on bioorganic chemistry, and in continuing our previous lecture on enzymes, we will be today studying enzyme regulation. Uh, Enzyme regulation is basically the regulation of differently enzyme catalyzed reactions, that how the rate of reaction in case of enzyme catalysis is regulated. That is called as enzyme regulation. And a connection exists among the products formed by different metabolic pathways from a common metabolite. Means we know that in case of biosynthetic reactions, a series of reactions they are controlled by a metabolic pathway, and these metabolic pathways they start from a common metabolite. For example, glucose is a common metabolite which can either be converted by oxidation to carbon dioxide, or it can be polymerized to glycosines, lipids, or non-essential amino acid and other sugar molecules. So all these steps leading to the conversion of a metabolite through a metabolic pathway into a product has to be regulated, because we require a specific amount of each entity present in the body. For example, the amount of glycogen which is required in our body to store for different energy requirements, because of the fixed amount of any substance required in the body. That means production of these substances need to be regulated, and this regulation comes from the regulation through enzymes. So for example, the glucose supply of the body, that can be achieved either from diet or from breakdown of glucose. So conversion of glycogen into glucose, that means through the metabolic pathway it needs to be regulated based on the requirement of the body about this glucose molecule. Similarly, polymerization of the glucose can be produced from some amino acids or lactates, and these processes of glucose utilization and synthesis they need to be regulated, and this regulation of rate of reactions catalyzed by enzymes need to be regulated, and this is known as enzyme regulation. The regulatory enzymes, as I have told you, a metabolic pathway involves many enzymes functioning in a chronological or some distinctive arrangement to carry out a metabolic process, and that metabolic process, say conversion of A into F through these steps. Say first the substrate A is converted into an intermediate B, which gets converted into intermediate C, that then into D, then into E, till the formation of the final product, that is F. And each step of this metabolic pathway is catalyzed by some particular enzyme. Say conversion of A to B is catalyzed by enzyme E1. This conversion B to C is controlled by enzyme E2. C to D, E3, D to E, E4, and E to F, E5. So control of a pathway is achieved through modulation of the activity of only one or few key enzymes. So regulation is not required for all these enzymes E1, E2, E3, E4 or E5. So we may regulate any one or two enzymes in such a way that the overall production of this is regulated. So these enzymes which control the activity of different types of substrates into products, they are known as regulatory enzymes. So they generally catalyze first or an early reaction in the metabolic chain, and that early step is generally a rate determining or rate limiting chemical reaction, and this controls the overall conversion of A into F. So this will control the overall pathway of that metabolic process, and it may also catalyze a chemical reaction unique to that pathway, and that unique step of the chemical reaction is called as committed step. So for example, if conversion of A into F, the conversion of B to C is catalyzed by enzyme E2. Say this is the rate limiting step as well as committed step. So this is a unique step, and this will determine that how the formation of F from A is regulated. So basically it means that out of all the enzymes involved in a metabolic pathway, only one or two they will act as committed enzymes and regulatory enzymes. So those enzymes which catalyze the rate limiting step or the committed step of a pathway, they are said to be under regulation. So these are called as regulatory enzymes. For example, if we say this substrate gets converted into a product through these intermediates, and these are the three enzymes which are required for conversion of this substrate into this product. Now when the end product, we know that the amount of this product required is fixed. So when that amount has been formed through these conversions, that means now the production of this product needs to be regulated. So when the end product exceeds the steady state level concentration, so when the amount of product required has reached its end, that concentration of the product has been achieved, then it will inhibit the regulatory enzyme so that this reaction may be normalized. So then what happens is, when we have achieved the amount of product formed which is required to our body, then this enzyme E1 will be regulated in such a way that conversion of S substrate into this intermediate will stop when we have already achieved the amount of product required in this reaction. So this type of control is known as feedback inhibition. So this product has given us the feedback that the reaction is to be stopped now. So this will ensure a high degree of efficiency in utilization of substrates or materials, and as well as the energy which is consumed for conversion of this substrate into the product. So this on off switch is normally at the start of the pathway, as you have seen that conversion of this substrate into an intermediate A. Before this is achieved, a regulatory mechanism is involved. So this on off switch is normally present at the start of the pathway. So that means all the intermediate formed, they are not to be regulated. So only we need to have only this enzyme needs to be regulated. So this enzyme will be called as the regulatory enzyme, and this on off switch is often controlled by a small molecule, and small molecule is present often in the product of the reaction. So this pathway is called as negative feedback regulation. So because product has been formed in the required quantity, so this has given us the feedback that now this enzyme needs to be regulated. Now, how this enzyme regulation is achieved? There are different ways by which the enzyme regulation can be achieved. One is by controlling the concentration of the enzymes. If we control the concentration of the enzymes, then we can synthesize the control of synthesis of various types of products. So by increasing the concentration of the enzyme, we can activate the process. By decreasing the concentration of the enzyme, we can repress the process. So like this, by changing the concentration of the enzyme, we can control the enzyme regulation. Then by degradation of the enzyme also we can control different types of enzymes. Then second type is by controlling the availability of the substrate, by changing the concentration of the substrate, the availability of the substrate, the production, degradation and compartmentation of the substrate can also lead to the enzyme regulation. Then we can use competitive inhibitors for increasing or decreasing the activity of the enzyme, or in other words, we can regulate the enzymes by reversible binding of the competitive inhibitor. When we require more of activity for the enzyme, we can remove the competitive inhibitors, then activity will increase. If we are to decrease the activity of the enzyme, we can add competitive inhibitors to that active site of the enzyme. This will decrease the activity of the enzyme. That is how the enzyme can be regulated in that way. Then controlling the activity of the enzyme. We can control the activity of the enzymes by binding modulators and effectors. These modulators, they can be negative modulators or positive modulators, similarly negative effectors or positive effectors. Positive effectors, they will increase the activity of the enzyme. Negative modulators or negative effectors, they will decrease the activity of the enzyme. So reversibly binding of these modulators and effectors, that will regulate the activity of the enzymes. Then reversible or irreversible covalent modification in the enzymes can also regulate their activity. So enzyme regulation can be achieved by different type of complexes. First of this category is multi-enzyme complexes. A multi-enzyme complex is where a series of chemical intermediates, they remain bound to the enzyme molecule as a substrate is transferred into the final product. For example, if this is the substrate and we want to get this product, and these are the three intermediates present, this multi-enzyme complex will bound all these intermediates on itself so that the transformation of the substrate into the product can take place. Example is pyruvate dehydrogenase complex. This is a multi-enzyme cluster which is isolated from Escherichia coli, E. coli, and this pyruvate dehydrogenate consists of different types of enzymes: pyruvate dehydrogenase or pyruvate decarboxylase. This is say E1, dihydrolipoyl transacetylase. This is another enzyme present in the multi-enzyme complex, and third is dihydrolipoyl dehydrogenase. So there are three multi-enzyme complexes present in the pyruvate dehydrogenase complex, and in addition there are five coenzymes present on this multi-enzyme complex. TPP, that is thiamine pyrophosphate, then lipoic acid, flavin adenine FAD, coenzyme A and FAD plus. So these are the five coenzymes which are also present on the pyruvate dehydrogenase complex. So all these enzymes and coenzymes, they catalyze different steps in conversion of a substrate into a product through these three types of intermediates, say, and all these they are present in the same pyruvate dehydrogenase complex, which is a multi-enzyme complex. So all these enzymes, they are present within the complex, and that is why the different steps of the metabolic processes, they are completed within the multi-enzyme complex, and in one or two steps those enzyme out of these they are regulated, and through these complexes enzyme regulation can take place. Another example of enzyme regulation by multi-enzyme complexes is through pyruvate dehydrogenase complex. So here we can regulate the enzyme activity through this multi-enzyme complex. You can see over here that pyruvate dehydrogenase triphosphate, three phosphate molecules, they are present, and when this reacts with water, then hydrolysis of this enzyme takes place, and phosphate dehydrogenase enzyme becomes active, whereas when these phosphate molecules, they are attached, this enzyme is an inactive molecule. So various enzymes, they are organized in such a way that the product of one becomes substrate for the adjoining enzyme. So for example, here we produce three phosphate molecules, and these three phosphate molecules, they can be used for the product formation. That is, when ADP is converted into ATP, then three phosphate molecules can be used, or when ATP is converted into ADP, then this active pyruvate dehydrogenase molecule will use up these phosphate molecules produced by degradation of ATP into ADP, and these three phosphate molecules, they will get attached to this enzyme, and this will change from active form to inactive form. So this is how the products of one step, they will be used as substrate for the other step. Similarly, the phosphate dehydrogenase phosphatase, when this is reacted in combination with magnesium and calcium 2 plus ions, then in addition, in presence of water, this will produce these three phosphate molecules by hydrolysis of this enzyme. So this is how enzyme regulation that can be used, that can be achieved by multi-enzyme complexes. Then another way of enzyme regulation is through pro-enzymes. Now pro-enzymes are those series of enzymes in which activation of the initial pro-enzyme by a biological signal activates the second pro-enzyme, which in turn activates the third, and it goes on till the whole metabolic process is completed. So for example, if we see this one, glycogen where the active site is having tyrosine, and when this uridine diphosphate glucose reacts with this enzyme, then hydrolysis of this glucose part is shifted from here to the tyrosine molecule, and this uridine diphosphate glucose becomes uridine diphosphate. So this is the structure for uridine diphosphate molecule. Then this will activate the this enzyme, and from here these glucose molecules, glucose 8 molecules of glucose, they get attached to this active site, and in the presence of glycogen synthesis, which is a in addition, this is having a branching enzyme present. This will again change the UDP glucose into UDP, and this is how these 8 glucose molecules, they will be polymerized to give us a polymeric compound, where alpha 1,4 and alpha 1,6 linkages of glucose, they will give us the product which is to be formed, and such type of process is regulated by these types of pro-enzymes. So the common example of such type of regulation is blood coagulation, dissolution of blood clots, then complement activation, and breakdown of glycogen, which is depicted over here. So this is the process of polymerization of glucose into glycogen, which is regulated by pro-enzymes. The next is enzyme regulation by isoenzymes. So many enzymes, they occur in several molecular form. So such enzymes which occur in several molecular form, they are called as isoenzymes or isozymes, and they are genetically determined, and they may be called as which are genetically determined, they are called as primary isoenzymes. So the general properties of these isoenzymes is that they have multiple forms of the same enzyme that catalyzes the same chemical reaction, and these can be differentiated from having different chemical and physical properties. The different molecular forms, they will have different chemical and physical properties, like electrophoretic mobility, kinetic properties, amino acid sequences, amino acid composition. All these, they will be different in the different molecular forms of the same enzyme. So the example of such type of isoenzyme is lactate dehydrogenase. This is present in wide variety of organisms, and there are 5 isoenzymes which are present on lactate dehydrogenase enzymes: LDH1, 2, 3, 4 and 5. These are the 5 isoenzymes. So that means lactate dehydrogenase have 5 molecular forms through which it can be represented, and all these 5 molecular forms, they will have different kinetic properties, different amino acid sequencing, and different amino acid composition in each of the molecular form, and each molecular form that can be separated by a cellulose acetate electrophoresis at pH 8.6. So we can differentiate these molecular forms from each other. Then another aspect is regulation of enzymes by zymogens. Zymogens are the enzymes which are synthesized by the body in an inactive form, and when they are synthesized, they do not possess any enzymatic activity. So they are synthesized in an inactive form, but these enzymes, they become active when one or more peptide bonds they are cleaved, leading to the conformational changes in the enzymes. So for example, if say this is the zymogen, and this is having different polypeptide chains. If say this polypeptide chain is removed from zymogen, then the substrate can interact with the active site of the enzyme. So otherwise you can say there was a lid which was stopping this substrate to interact with the active site. But once this lid is removed, the polypeptide segment is removed from the zymogen, the substrate can approach the active site of the enzyme. So we can have this enzyme an active enzyme when a part of the polypeptide chain is removed from this enzyme. So basically when this is removed, a conformational change occur in the enzyme which make it active for formation of an effective enzyme substrate complex, which lead to the product formation. And the activity of the zymogens can be achieved through different methods. For example, if we are having this enzyme pepsinogen, and this is an inactive form, and when this is treated with HCl, this gets converted into pepsin, which is an active form of the enzyme. So this is zymogen. When one part is removed or some conformational change occurs in presence of HCl, this gets converted into the active form of the enzyme. Trypsinogen, again in the presence of enterokinase enzyme, gets converted into trypsin. This is an inactive form, this is an active form of the enzyme. Prothrombin, in presence of thrombokinase and calcium 2 plus ions, it gets converted into thrombin. This is an active form of the zymogen. This is zymogen. Then pepsinogen can also be converted into pepsin by reacting with another molecule of pepsin. This will make it active. So these types of activation of enzymes takes place due to the conformational changes in the enzyme due to the cleavage of different peptide bonds. Then enzyme regulation can also be achieved through covalent modification. The enzyme activity can either be increased or decreased if we use covalent modification of enzyme regulation. And some common covalent chemical modification, they are used in phosphorylation and dephosphorylation, or acetylation or deacetylation, methylation or demethylation. So if we see this reaction, so this enzyme is having serine as the side chains of the enzyme, and when serine is present in the side chain, the enzyme is not in the active form. This is less active form of the enzyme, and when this is reacted with the two molecules of ATP in presence of enzyme protein kinases, then the phosphorylation of this serine side chain will take place and the enzyme will become more active. So reaction of this enzyme with ATP will lead to the formation of more active form of the enzyme. Similarly, this more active form of the enzyme can be converted into less active form of the enzyme in the presence of enzyme phosphoprotein phosphatase enzyme will phosphatase will convert this enzyme into less active form. So here dephosphorylation will take place, whereas in the first case phosphorylation has taken place. So the activity of the enzymes, they can be regulated by covalent modification, which you have seen either through phosphorylation or through dephosphorylation. So you can see over here this OH has formed the bond OP bond, which is covalent in nature. That is why this is called as covalent modification. Then another example of covalent modification is in the enzyme regulation, glycogen phosphorylase enzyme is used for the conversion of glycogen into 6 glucose phosphate and glucose 1 phosphate, and depolymerization of the glycogen will take place. So hydrolysis at this 1,4 glucosidic linkage will take place, and one monomer of the glucose will be hydrolyzed to get glucose 1 phosphate in presence of enzyme glycogen phosphorylase. So basically the hydroxy group will undergo covalent modification with the addition of this phosphate molecule, and we will get glucose 1 phosphate, and this will either increase the activity or decrease the activity of different types of enzymes. But here in this particular case the activity of glycogen phosphorylase is affected. Then is allosteric enzyme regulation. The enzymes in metabolic pathways whose activities can be regulated by non-covalent interactions of some compounds at sites other than the catalytic sites or active sites of the enzymes, they are called as allosteric enzymes. So when the activity of the enzyme is affected due to the presence of sites other than the catalytic sites or catalytically active sites, those enzymes, they are called as allosteric enzymes, and those sites, they are known as allosteric sites. The allosteric enzymes, they usually rate determining enzymes, and they play a significant role in control and integration of the metabolic processes. So if we want to increase the rate of the metabolic process or decrease the rate of the metabolic process, that will be achieved through these types of allosteric enzymes. The allosteric means other type of enzyme. So allosteric site is a unique section of an enzyme which results in catalysis. So catalysis, rate of catalysis in these types of enzymes, that will be controlled by the sites which are called as allosteric sites, which will either activate or deactivate the catalytic sites present in the enzyme. So this can be achieved through changes in the shape of the enzyme, and these types of regulations, they are non-covalent interactions. So these are because of non-covalent interactions at the allosteric sites, and these allosteric sites, they are affected because of the particular ligands which are known as effectors, modulators, or modifiers. So these effectors, modulators, or modifiers, they are responsible for enzyme regulation through allosteric site. So for example, if this is the molecule, say this is the regulatory site, say this is the active site of the molecule, and this is the substrate which is to interact with this, and this will be controlled by this regulatory molecule present on the allosteric site. This regulatory molecule will be a factor, modulator or modifier, and this will control the activation or deactivation by changing the shape on the active sites of the enzyme. This will lead to the more reaction or less reaction, activation or deactivation. So you can see over here the shape of the active site becomes same as that of the substrate, whereas here the shape has changed in such a way that these substrate molecules, they cannot be accommodated in the active sites. So this is what the regulatory molecule will do, and regulatory molecule is a factor, modulator or modifier. So this is how activation or deactivation will take place through allosteric enzyme regulation. And then allosteric enzymes, they are mostly oligomers consisting of two or more polypeptide chains or subunits known as protomers. And these protomers, they will have the allosteric site present on that. And allosteric enzymes, they will show two types of interactions: homotropic interaction or heterotropic interaction. In case of homotropic interaction, you will see that here the same ligand will influence the positivity toward cooperation between different modular sites. So same ligand will determine the activity or deactivity of the enzyme. Whereas in case of heterotropic interaction, the interaction will involve the effect of one ligand on the binding of the another ligand. So here only one ligand will be involved, here different ligands they will be involved.
For example, in homotropic allotropic interaction
will be responsible for the activity of other ligands. Whereas here in case of heterotropic allosteric enzyme regulation, only one ligand will be responsible for the activity of the enzyme. Now there are two theoretical models for allosteric enzyme regulation, and these two models, they are concerted or symmetry model, or second one is sequentially induced fit model. And in both the cases of these models, these are the two assumptions which have been made. That first assumption is that each subunit of the oligomeric protein exists in two forms. One is T form and other is R forms. T form is the form where the binding of the ligand is with low affinity, whereas R form of the subunit is where the binding of the ligand will be high affinity binding. So this is the assumption number one. And second assumption is that conversion of T into R or R into T. So these transformation, low binding to high affinity binding or high affinity binding to low affinity binding of the ligand with the oligomeric proteins will involve non-covalent bonds. So these interactions, they will be non-covalent interactions, and these non-covalent interactions, they will lead to the change in quaternary structure of the enzyme, and because of the change in quaternary structure of this enzyme, there will be an enzyme regulation. The first model, the concerted model, was given by Monod, Wyman and Chenguish, and this is also known as all or none model. So this is called as all or none model because according to this model, the allosteric protein can exist only in two states. So either that state can be T or R. So either the protein will exist in the low affinity binding mode or in the high affinity binding mode. So that means the oligomer sections, they can have either TT or RR conformation. It is not possible that in the same allosteric protein the two subunits, one subunit is having T, that is low affinity, and other is having R, that is high affinity. So TR or RT conformation is not allowed. The conformation can either be TT or RR, that is either low affinity binding of the two subunits or high affinity binding of the two subunits. So this is the basis of this concerted model. So here this is responsible for kinetic behavior of many allosteric proteins, but it cannot account for negative cooperativity. That is decrease in affinity for a ligand by allosteric protein as the site becomes occupied. So this concerted model can be shown like this. These are the two subunits of the enzyme, and these are the two active sites, and you can see this shape of the substrate and shape of the active site. They are not in conformity. So the active sites, they have the unfavorable conformation to accommodate the binding of this substrate. So in addition to these active sites on the subunits, we are having these allosteric sites present, and these allosteric sites with the help of a factor or modulator will change the shape of these active site in such a way that the substrate can form a stable enzyme substrate complex with the protein. So a negative effector or modulator will preferentially bind to the T form. T is the uh, low binding state, and allosteric protein will become in the TT conformation. So this is the TT conformation of the allosteric protein. So both the subunits, they have low affinity for the substrate. So binding is possibility of binding is very low because of the shape of these active sites, and such a factor will be known as a factor or modulator will be known as negative factor or modulator, and cooperativity will also be negative cooperativity. Similarly, positive factor or modulator will change the shape of these sites in such a way that the state of the subunit will become R, that is high affinity state will be formed. So you can see over here the shape of the active site has changed. Now this active site has a favorable conformation for binding of the substrate, and this is RR state, that is high possibility of the binding of the ligand. So these are some of the allosteric modulators which are used in different enzymes. For example, in glutamate dehydrogenase, the allosteric activator used is ADP, and allosteric inhibitor is ATP or NAD in combination with NADH. So that means if ADP is present, the activity of the enzyme will increase, and if ATP is present, the activity will decrease. This will inhibit the allosteric interaction, this will activate the allosteric interaction. Similarly, hexokinase enzyme, ADP is activator, ATP and glucose 6-phosphate, they are inhibitors. Isocitrate dehydrogenase, this is activator, this is inhibitor. In pyruvate carboxylase enzyme, acetyl coenzyme is activator, allosteric activator, ADP is allosteric inhibitor. The next is sequential model. So this was given by Covalent, and he proposed that ligands bind to a subunit, then a conformational change is induced which stabilizes the ligand allosteric protein complex in such a way that the second ligand molecule is bound more readily than the first. So that means the binding of the ligands, the rate at which the binding of the ligand will take place will increase if this conformational change is achieved through positive cooperativity. So when binding of the second ligand, rate of binding of the second ligand is faster than the binding of the first, then this is called as positive cooperativity. And if the binding of the second or additional ligand molecule becomes difficult, then this will be called as negative cooperativity. So this model can be represented like this. For example, if these are the two subunits of the enzymes, then these are the allosteric sites, and this means when the shape of these active sites is not in conformation with the substrate, the conformation is unfavorable. That means the allosteric enzyme is in the TT state. And if positive factor or modulator preferentially bind to the R form and maintains the allosteric protein in the RR conformation, this is the RR conformation. So here the allosteric factor or modulator has bound to the allosteric site, and the shape of this active site has become favorable, which will lead to the binding of the substrate molecule. Similarly, negative factor or modulator will bind to the T state, and this will maintain the allosteric protein in the TT conformation. So this is TT conformation. So that means here the negative factor or modulator has bind in the T state. This is RR state. Here the positive factor or modulator have bound in the R form, and this maintains the allosteric protein in RR conformation. And in case of sequential model, one F factor or modulator can change only one active site of the molecule, active site of the subunit become favorable, other remain unfavorable. So such a state is called as TR state. So one is with high affinity, other is with low affinity. This is T state, this is R state. So here the positive cooperativity goes on decreasing, whereas negative cooperativity goes on increasing. So this is all about enzyme regulation. Thank you very much.